नमस्कार विद्यार्थी मित्रांनो, आज आपण समजून घेणार आहोत स्ट्रक्चर ऑफ अँटीबॉडी immunoglobulins. So first of all, what is antibody? Antibody is a glycoprotein which is highly specific to antigen, means foreign body. Simply we can say antibodies or immunoglobulins are glycoproteins and they are very specific for a particular antigen. Antibodies are produced by lymphocytes, B lymphocytes. These are our immune cells. We already studied our immune cells, B lymphocytes and T lymphocytes. So B lymphocytes produce the antibodies. When any antigen enters in our body, or you can say due to antigenic stimulation, B lymphocytes get sensitized and these sensitized B lymphocytes, they produce their clone cells, to which we call plasma cells. And these plasma cells, they produce antibodies. So antibodies are produced by plasma cells, which are actually the clone cells of B lymphocytes. A mature plasma cell can produce about 2000 molecules of antibodies per second. We have to study the structure of antibody. Typically, antibody is Y-shaped molecule consisting of 4 polypeptide chains. Polypeptide chains means chains of amino acids. Out of these 4 polypeptide chains, 2 chains are heavy chains and 2 chains are light chains. So this is heavy chain, another heavy chain. So these 2 are heavy chains. These are the heavy chains of amino acids or polypeptide chains, and 2 light chains are also present. So now these are the light chains. This is first light chain and this is second light chain. So in all there are 4 polypeptide chains. This is light chain, you can call it L chain, and these are H chains, heavy chains. So there are 4 polypeptide chains. Both these heavy and light chain have 2 regions, variable region and constant region. In this diagram, now you can see I divide each polypeptide chain, both the heavy chains and both light chains into two sections. So this is the variable region of light and heavy chain, and the remaining portion, this one, is constant region of light and heavy chain. Variable region is also called antibody binding site, or it is also called paratope. This region, variable region, it varies depending upon the structure of antigen. I would like to make some changes in variable region of light and heavy chain. So I made some changes. You can note those changes. See now the change in the variable region or paratope. The structure of paratope or variable region depends upon the structure of antigen for which that antibody is formed. Suppose this is antigen, any foreign body. On the surface of this antigen, there are some specific areas to which we call epitopes. Epitopes. These structures are epitopes. This is epitope on this antigen. This one is antigen. And on the surface of that antigen, epitopes are there. Look the shape of the epitopes, right? I would like to draw another antigen here. Suppose this is another antigen and these are the epitopes. Now also you observe the shape of the epitopes in the second antigen. So these epitopes and these paratopes of the paratopes are present on antibody and epitopes are present on antigen. They are complementary to each other, and depending on the shape of the epitope, the shape of the paratope or variable region of the antibody depends. और इपिटोपचा आकार कसा आहे, इपिटोपच्या आकारानुसार हा जो पोर्शन आहे त्याच्यामध्ये बदल होत असतो आणि म्हणून या पोर्शनला आपण व्हेरिएबल रिजन असं म्हणतो किंवा अँटीबॉडी बाइंडिंग साइट असं म्हणतो किंवा पॅराटोप असं म्हणतो. ऑल these four polypeptide chains are held together by disulfide bonds. So there are 4 disulfide bonds. These are the 2 disulfide bonds and one is present here in between the constant region of light chain and constant region of heavy chain. Another one is present on the other side and 2 are present in between the heavy chains. So there are 4 disulfide bonds and these disulfide bonds hold all these polypeptide chains together, and this is how the structure of antibody. Next topic is classification of antibodies or types of antibodies. Antibodies are classified into 5 classes. So first we will write down the class or type, then about the structure, then about the location where that antibody is mainly found, and then about the function of that antibody. The first type is IgG, immunoglobulin G. It is monomer type of antibody. Structure of this antibody is like monomer. Monomer means the structure consists of single Y-shaped structure. This antibody is mainly found in blood, lymph and extracellular fluid. This antibody IgG is the only transplacental antibody, means only this antibody can pass to the fetus, from mother to the fetus through placenta, and it provides natural passive immunity to the fetus. Second class is IgA, immunoglobulin A. This is structurally dimer type of antibody. Dimer means it consists of two Y-shaped structures. Di means two. It is found in saliva, tears, milk, respiratory and intestinal secretions, and it provides local mucosal immunity. It provides local mucosal immunity. The next type or next class is IgM, immunoglobulin M. This is the largest in size. Why it is largest? Because structurally it is a pentamer, means made up of 5 Y-shaped structures. It is found in blood and lymph. And another one more point about the IgM you should remember, that is after infection the first antibodies which are formed, those are nothing but an IgM. So after infection first antibody formed after infection, जेव्हा कोणताही अँटीजेन आपल्या शरीरात जातो त्यावेळेला सगळ्यात पहिले ह्या अँटीबॉडीज तयार होतात, IgM. And another thing you should note about the IgM type of antibodies, that is they are based at agglutination. Agglutination means they bound to the antigen and they make it immobilize, and they enhance the phagocytosis, engulfing process of that antigen, and that is another important function of this antibody. Next one is IgE, immunoglobulin E. It is a monomer type of antibody, means made up of single Y-shaped structure. It is bound to mast cells and basophils. It is attached to the mast cells and basophils. Mast cells are present in the connective tissue and basophils are present in our blood, right? And this antibody plays important role in allergic reactions. The function of this antibody IgE is very important in allergic reactions. And the last one is fifth class is, or fifth type of antibody is IgD, immunoglobulin D. It is again monomer type of antibody, means made up of single Y-shaped structure, and its location is on the surface of immature B lymphocytes or B cells. It acts as a B cell receptor for antigen recognition. Means this antibody IgD plays very important role in antigen recognition. It helps the immune cells to identify, to recognize the antigen. And for that purpose, this antibody IgD get attached to the surface of B cells. Next topic is antigen antibody complex, which is also called immune complex. Antigen antibody complex or immune complex is produced due to binding of antibody to the specific antigen. When antibody binds to specific antigen, then a complex is formed, and that complex is called antigen antibody complex. On the surface of antigen, specific proteins or polysaccharides are present, and these specific proteins or polysaccharides which are present on the surface of the antigen, those are called epitopes. Suppose this is antigen, and on the surface of this antigen, whatever the proteins or polysaccharides are present, those are called epitopes. Similarly, in the variable region of polypeptide chains of antibody, paratopes are present. So, when antigen enters in our body and against that antigen specific antibodies are produced, what happens? These paratopes of antibody binds with epitopes of antigen in a lock and key mechanism, and it forms antigen antibody complex or immune complex.